So just a general outline. So I'm going to focus initially on the first part about general study design recommendations for comparative clinical endpoint studies for orally inhaled products. And in the second half of my presentation, I'm going to present common deficiencies and helpful recommendations for comparative clinical endpoint studies and their study design. So just as a reminder, I know Dr. Conti went over this in her presentation, for why a comparative clinical endpoint study is needed for these types of products is the site of action is at the lungs. And so there's uncertainty regarding the correlation of in vitro and in vivo PK data to the clinical effects at the site of action. And so that's why a comparative clinical endpoint study is needed for these types of products, and a pharmacokinetic study is not enough on its own. So for those of you who are unfamiliar with the basic study design of a comparative clinical endpoint study for these types of products, they're conducted in patients with asthma or COPD. There's a placebo run-in period. The primary endpoint is based on spirometry, such as forced expiratory volume in 1 second, or FEV1. And their product-specific guidance usually recommends rescue medication. And we'll talk about how that's important when it comes to study design. So in terms of general study design recommendations when thinking about designing your study, follow the recommendations in the product-specific guidance, as this represents the agency's current thinking on how best to conduct the study. Differences from the product-specific guidance may be acceptable, but require justification and must be pre-specified. And I also want to emphasize that these differences should be included and documented in your original ANDA submission. Do not wait for an information request to be sent out, because we will be looking for this information to justify the differences. And also, as you've heard in other talks, we recommend that you discuss the comparative clinical endpoint study in the pre-ANDA process. And so I'm going to provide a number of examples of differences from the product-specific guidance that you should provide justification and you may want to discuss in the pre-ANDA process. And that includes any differences in the study population compared to the product-specific guidance recommendation, including age, non-US population, or any differences in the reversibility criteria for any studies involving asthmatic patients, treatment duration, any differences in statistical analysis, and this includes the baseline or the primary endpoint difference, study population differences, any differences in the retention sample, and I'll talk a little bit more about that in the process. So I'm now going to talk about 8 common deficiencies and provide helpful recommendations and tips to improve and consider when conducting your study design. And these deficiencies also apply to a lot of comparative clinical endpoint studies for complex products, not just those for orally inhaled products. So I ask that you pay attention and consider that while listening to the rest of the conversation and discussion. Including study drug compliance, out-of-window study visits, bridging information, and also retention sample process. So the first deficiency and helpful recommendation revolves around prohibited concomitant medications. And so the first recommendation is to use the product-specific guidance. The product-specific guidance outlines the products that you should consider and further evaluate if subjects take during the treatment period. But also to consider other concomitant medications that may affect evaluation of the primary endpoint, such as montelukast, ipratropium. The medications that are listed in the product-specific guidance are not meant to be an exhaustive list. And so if a subject or a patient in the study is taking a medication that is indicated for asthma or COPD, it's important that you look further at those medications, the timing, the duration, and consider and make an evaluation of whether or not that subject should be included in the per protocol population. But again, the important thing to realize is the medications listed in the product-specific guidance are recommendations, but they're not meant to be an exhaustive list. Along with the concomitant medications, it's important that you let us know that any medications that are listed in the concomitant medication data set specify if the subject actually used the as-needed or PRN medication. Obviously, we're interested to know if subjects actually took it If it was just an as-needed medication, but the subject or patient did not take the medication, that would affect our evaluation. And so, as I mentioned, rescue medication is an important part of the study design, and I'm going to provide some recommendations to help and for you to consider when designing your study. So it's important that you pre-specify the rescue medication use, and that includes a number of things that I've listed above. And that includes the maximum allowable amount of rescue medication use in what time period, any limits on rescue medication use. For example, if the rescue medication use is prohibited from being used within 6 hours of the primary endpoint evaluation, and how subjects who exceed maximum allowable amount of the rescue medication in the comparative clinical endpoint study will be handled in the statistical analysis. So furthermore, it's important to realize that we must control for rescue medication to avoid confounding variables. And so, as I mentioned, most of the product-specific guidances provide recommendations on the washout period or time that the rescue medication use cannot be taken before spirometry evaluation. And we ask that you consider those recommendations in designing your study. The other thing that I wanted to emphasize and point out is that subjects who use rescue medication within 6 hours, if that's what you decide to include in your study design, before spirometry evaluation, that is used for inclusion and exclusion criteria and primary endpoint evaluation should be excluded from the per protocol population. And so it's not just those subjects who use rescue medication within 6 hours of when a primary endpoint evaluation was taken place, but also if the inclusion or exclusion criteria was measured at a certain visit, if the subject used the rescue medication within 6 hours, that also would affect the evaluation of that inclusion and exclusion criteria, and therefore you should look further and evaluate, and those subjects should be excluded from the per protocol population. So we recommend that you provide a rescue medication data set that includes the date and time of each use of rescue medication for each subject. And I've provided this as an example. The important thing to note is that each time that a subject used rescue medication, it's listed as a separate row. So you'll see for the first subject, there's actually 3 listings, and those are all separate entries for different times that they use rescue medication. And so we want to be able to evaluate and look and ensure that subjects did not use the rescue medication within the prohibited time period as specified in the study protocol. And also, if you do decide to include rescue medication use in the concomitant medication data set, we ask and recommend that you clearly document which listings, which rows are rescue medication use. So now I'm going to transition to treatment failures. So these are subjects who are discontinued due to a lack of efficacy. And it's important to realize that by having a definition and creating this into your study design, it ensures subjects with the worst case scenario are included in the statistical analysis and to avoid potential bias. And again, it's important to pre-specify this in your study protocol, and that includes the definition, how these subjects are handled for the statistical analysis, whether they're included or excluded, and that includes the per protocol population or the modified intent to treat population. And again, how they're being handled in the actual statistical analysis, whether a last observation carried forward procedure is going to be used or something else. And so on this slide, I'm going to talk about the definition and things to consider when creating your definition of treatment failures. So think about those subjects, look at their reason for discontinuation. For example, for a study for asthma patients, if a subject discontinued due to asthma exacerbation or lack of efficacy, they should be considered and likely be considered a treatment failure. For those subjects who had an adverse event that led to discontinuation from the study of asthma exacerbation or worsening of asthma. And also those subjects who required alternative therapy, such as rescue medication use above the maximum allowable amount, or other prohibited concomitant medications, such as corticosteroids, should be considered and looked at further when creating your definition and study design for treatment failures. And again, it's important to realize that when you do create your definition for treatment failures, to include that subjects must complete at least a certain amount of days of weeks of treatment with the study drug. So for example, if a subject discontinues on day 1 or day 2 of a study due to asthma exacerbation, they probably haven't had enough of the study medication for it to take effect. And so including the must complete at least a certain amount of days of treatment with the study drug is needed to ensure that the effects of the study drug would be noted. And again, we leave that up to the applicants, but in terms of recommendations on how to decide how long of a time period, and this varies tremendously based off the study and the study drug, it's how long you would expect for the medication to take full effect. So another deficiency I'm going to talk about is the percent predicted FEV1. And this ensures subjects have the disease of interest and consistent criteria is being used to determine the inclusion criteria. The thing that I want to emphasize about this is to provide detailed information, including the exact formulas used to determine the predicted FEV1. So these formulas should be pre-specified and the same for all clinical sites, and they should be included, again, in the study protocol. So I'm going to provide 2 recommended approaches. So, for example, certain study protocols will say something like the percent predicted FEV1 for inclusion criteria is based off of N. Haynes 3, Hankinson 1999. But this doesn't provide us enough detailed information, as this leaves some room for interpretation at each of the clinical sites. And so what we would like to actually see is the actual formula, an example I provided, so that we know that each clinical site used the exact specific criteria. A second recommended approach is you can preload the same formulas into spirometry machines and provide all clinical sites with sponsor-provided spirometry machines. But the important thing to note is you still need to provide the actual formulas used in your study protocol and your study report to ensure that all clinical sites use the same criteria. And again, this limits the for interpretation by different clinical sites. So another area I want to clarify and hopefully provide some guidance is on study drug compliance calculation. And it leads, sometimes we've seen some improper calculation due to the expected doses and study drug compliance. And just for those of you who are more familiar, the study drug compliance is the number of doses received divided by the number of expected doses. And so I just want to clarify about the denominator, the number of expected doses, and emphasize this is based on the study design. And so this should be a same number for all subjects. It does not change if the subjects discontinue from the study early. So it does not matter, or it's not affected when subjects discontinue or how long they're in the study. And we do understand why there may have been some confusion or different interpretation. But again, that's why we're talking about it now and emphasizing that this should be the same number for all subjects and is not affected if any subjects discontinue from the study early. And I'm going to go over an example that will hopefully emphasize this point a little bit better. So for example, if a subject received 18 doses of a study drug and they were discontinued from the study on day 11, and the study duration for this treatment was 28 days, and the study drug is taken twice daily. The study drug compliance, again, here's the formula. The number of expected doses, so this is the denominator, is again based off the study design, which is 56 doses, 2 doses per day times 28 days, not 22 doses, 2 times 11. And so there's the formula, and so that would be the correct study drug compliance calculation for this subject. Another recommendation I want to help clear up is out-of-window study visits. And so windows for all study visits should be pre-specified in the study protocol. And this is an example taken from not an ANDA, actual ANDA, but you can see that the visit windows are plus or minus 2 days. It's important that these are pre-specified and that whatever is pre-specified should stay constant and consistent and be used in the statistical analysis. They should not change after the study has begun. I just want to point out and recommend that subjects who do not complete the visit in which a primary endpoint is evaluated within the pre-specified visit window should not be included in the per protocol population. We understand that sometimes sponsors would like to include as many subjects in the per protocol population, but it's important that subjects who complete the visit in which a primary endpoint is evaluated outside of the visit window should not be included. But subjects with out-of-window study visits for interim visits that do not contribute to a primary endpoint evaluation may remain. And so, for example, if there's an asthma study that's 4 weeks long and the primary endpoint is evaluated at week 4 visit, and there is a scheduled in the study design at week 2 where there's just a telephone visit where the subjects just check in with the clinical site, but there's no spirometry evaluation. If subjects have an out-of-visit window for week 2 visit, that's okay because there's no spirometry. That would not affect the primary endpoint evaluation. But if they complete the visit at week 4 out of the pre-specified window, they should be excluded from the per protocol population. So the last 2 things I'm going to talk about is bridging information. And this is when there's changes in formulation, manufacturing, device, and we realize this is going to happen during the process. And some examples include filling overage, blending process time. To avoid this, we recommend that you use the to-be marketed or the commercial drug device product in the comparative clinical endpoint study. So we ask and recommend that you provide a list of all differences in formulation, manufacturing, and any device component for the drug device used in the comparative clinical endpoint study compared to the to-be marketed commercial product and the other pivotal studies submitted in support of bioequivalence. And you should provide this in the original ANDA submission. If you do decide to do any justification or bridging information, we ask that you provide that justification in the original ANDA submission for all differences. And also, as others have said, recommend to discuss major differences in the pre-ANDA program. The last thing I'm going to talk about are retention samples. We recommend that you follow the CFR, the applicable regulations, and FDA's guidance for industry on handling and retention of BA and BE testing samples. I just want to point out that there's a requirement for quantity, particularly important for the endpoint studies, that there's a recommendation and requirement to retain reserve samples for every shipment to all clinical sites, and this includes all subsequent shipments, and obviously follow the other requirements as listed. And we realize for comparative clinical endpoint studies, there's a potential for large number of shipments and large number of clinical sites, but want to emphasize that all aspects of the guidance and regulations still apply. And that's a copy of it for those of you who are not as familiar. So for the retention sample, we recommend that you develop a retention sample process and procedures early on in the design of the comparative clinical endpoint study. You discuss any alternative approaches in the pre-ANDA program. And remember that the goal of this is it's important to ensure study integrity and public's trust in generic drugs. So for summary, for my part of the presentation, use the product-specific guidance, discuss alternative approaches and significant differences in the pre-ANDA program, and justify differences from the product-specific guidance with adequate and scientifically sound justification, and provide it in your original ANDA submission. So here's some references, and I thank you for your time and attention.
Thank you, Drs. Ma and Spagnola.